Good evening, everyone. Welcome to the Wall Exchange. My name is Janice Sarah, and I'm the director of the Peter Wall Institute for Advanced Studies at University of British Columbia. The Peter Wall Institute is dedicated to advancing innovative and fundamental research that can generate new ideas, new thinking, and ultimately make important contributions to society. And the Wall Exchange this evening is part of a series of downtown lectures sponsored by the Peter Wall Institute. Its objective is to help us collectively engage in conversations about important and timely issues within our global society. And certainly tonight's conversation promises exactly that. I'd like to thank the Georgia Straight magazine for co-sponsoring this event, and to thank CBC Radio 1 Ideas program producer Yvonne Gall and her staff for taping the event for a future CBC Ideas program. The program this evening, you've already heard the sounds of the marvelous Oscar Hicks Jazz Sextet. Our sincere thank you. Let's give them a round of applause. So the rest of the program, Dr. Brett Finlay will speak for about 50 minutes, followed by a 40-minute question and answer period. And we're very delighted and honored to have Michael Serapio, who's the national morning anchor of CBC News Network in Toronto, who's going to serve as moderator of the Q&A period. Michael Serapio began his career as a producer, doing the parliamentary news programs that we've all come to love. He was twice nominated for a Gemini Award for this work. He also hosted a news magazine show in 2001 for Pride Vision Television, and he subsequently worked on a series of news journalism roles for CTV, joining CBC in 2010. And as I mentioned, he's now the national anchor in Toronto for the morning show. Thank you, Michael, for your willingness to serve this evening. Behind me on the screen, you can see our Twitter handle and the hashtag #WallX. And so in addition, at the question and answer period, in addition to the microphones that are going to be at the front, we'll be taking questions tonight via the hashtag. And you can send your questions in during the lecture as things occur to you, or in the first 2 minutes after the lecture ends. And so Michael will ask some questions of those of Dr. Finlay during the question and answer period. Now to introduce Brett, who is a friend and colleague. Dr. Finlay is a distinguished professor at the Peter Wall Institute for Advanced Studies. He's also a professor at the Michael Smith Laboratories and the departments of biochemistry and molecular biology and microbiology and immunology at the University of British Columbia. Dr. Finlay's research interests are focused on host pathogens interactions at the molecular level. By combining cell biology with microbiology, he's been at the forefront of an emerging field called cellular microbiology, making several fundamental discoveries in the field and publishing over 400 papers. Among his many achievements, Brett led the SARS Accelerated Vaccine Initiative, which developed 3 SARS vaccine candidates within 6 months, and I believe he actually beat the Center for Disease Control in the United States by just a few days. He developed a cow vaccine. He developed a cow vaccine for E. coli 0157, the pathogen that caused the outbreak, the major outbreak, we'll remember, in Walkerton, Ontario. And he proved the concept that animal carriers of pathogens can be vaccinated to prevent human disease. We could actually wipe out E. coli 0157 now if we had the political and economic will. Of tonight's lecture, Bugs or Us, the role of microbes in health, disease, and society, Dr. Finlay has said we are entering a golden era of our understanding of microbes. New technologies give us unprecedented insights into health and disease. Dr. Finlay is an officer of the Order of Canada and the Order of British Columbia. He's received numerous scientific achievement awards nationally and internationally, and moreover, as you've just seen, he's an accomplished musician. Please join me in welcoming Brett Finlay.
Okay, get scrapped up here. My Britney Spears act. Okay, well, good evening. Thank you, Janice. That was a very kind introduction. I appreciate that a lot. I'd also like to thank the Peter Wall Institute for sponsoring these events where we get to explain what we do to the public. I think they're really important. And also a personal thanks to Peter Wall himself, who for the last 12, 13 years is pushing me to expand my horizons and giving me the freedom and the opportunity to do that. So tonight we're going to enter an invisible world. Maybe invisible, but it plays an important part of our life and impact on us. And that's what I want to do. I want to tackle the idea of microbes and how they impact on us. So I will warn you, I have a perverse sense of humor. We're going to talk about feces, and so this lecture is restricted for fecal contents. And if you don't like it, tough. There will be fecal content. So, where do we start our story? Well, we've got to go back close to 350 years when Antti van Leeuwenhoek made his very first microscope. And he figured, well, what do we look at now? So he swabbed out the inside of his mouth, put it in his fancy little microscope, and looked up to it. And lo and behold, he saw all these little things. He called these things animalcules. And he made the stunning observation that there are more of these little things in his mouth than all the people in the United Netherlands, which is Holland, where he was living at that time. And that was really the first time we have ever seen and encountered microbes. And that was where the story begins. So we have to enter this invisible world of microbes. And when I say microbes, I mean these are bacteria, viruses, fungi, and other single-celled organisms. The thing they have in common is they're all invisible to the naked eye. So viruses are the very tiniest of things, and you can only see these if you have a very specialized electron microscope, incredibly tiny. They can't divide on their own, viruses. They need other cells to infect to do this. So the smallest free-living form of life are bacteria. And these things are still extremely small, but they have the capacity to grow on their own. So the bacteria are generally 1 micron, which is 1/1000th of a millimeter. And the tiniest speck an eye can see is about 0.1 of a millimeter. So you'd have to take at least 100 of these bacteria lined up end to end, see the absolute tiniest speck the human eye can see. So there's several other kinds of microbes, as I've said, single-celled, eukaryotic cell, etc. And these are a little bit bigger, but again, they're all invisible to the naked eye. Okay, so we discovered microbes. What came next? Well, next came the microbe hunters. And here are two famous gentlemen, Robert Koch and Louis Pasteur, that are probably two of the most famous microbe hunters. And this happened in the late 1800s. And up until then, nobody knew what caused infectious diseases. They had all these crazy theories about swamp water and bad gas and air and all these other things, and no one really knew what caused these things. Many false type descriptions. Then these two guys came along, and they had a profound impact because what they showed is that microbes cause disease. Robert Koch showed that the bacteria that causes tuberculosis, the ones that cause anthrax, and the ones that cholera, 3 major human diseases, were all transmitted by microbes. And not only do that, he isolated and grew these microbes in the very early days. Based on this, he set up a set of rules called Koch's postulates about how you actually prove a microbe causes disease. And to this day, we follow these rules when we basically try and prove a new disease is caused by a particular microbe. Louis Pasteur was his contemporary and also his competitor, and these two had a real go at it in terms of discovering microbes. And Louis Pasteur really confirmed what we call the germ theory. He really proved microbes cause disease. He developed the first vaccines to rabies and anthrax, and we know Pasteur most famously for pasteurization, heating of things to kill the bacteria. That all came from Louis Pasteur's work. So microbes cause disease. And if you read history, you realize that microbes have had a profound impact on human history. Going back to the Dark Age, the middle 1400s, there was bubonic plague caused by a bacterium swept across Europe. It killed 1/3 of all the people in Europe then. Napoleon, when he decided to take on Russia, his major military campaign took all the soldiers into Russia. He lost. It wasn't the Russians that defeated him, it was dysentery, another bacterial diarrhea-type disease. And this disease just swept through his ranks. They were malnourished and poorly supplied, etc. And only 1/5 of his entire soldiers came back from the campaign. The disease wiped that many out. In the U.S. Civil War, for example, more people died of infections than bullet wounds. That was pre-antibiotics, but obviously a major impact. And in the early 19th century, in 1918, there was what we called the Spanish flu, or the big influenza outbreak. This swept around the world, infected up to half the world's population, and actually killed more people than those that died in World War I at the same time. It's estimated up to 40 million people died of the flu, and only 20 million people died in World War I. So huge impacts on human history. More recently, we see lots of examples. One example that Janice alluded to was SARS. And we experienced SARS in Canada a lot in 2003. And as you know, that pretty much shut down Toronto and had numerous other effects. And it's estimated that SARS actually cost the world over $90 billion, as well as all the other inconveniences of filling out forms when you're customs. Do you have a fever? If so, go into a very dark room and don't come back, and all these other things. So, what do these things do? Well, they still kill an awful lot of people in infectious diseases. They cause about a quarter of all the world's deaths. About 17 million people, a quarter of the people that die worldwide, die of these infectious diseases. And you say, oh, well, that's just developing countries. We're never impacted by it. Wrong. This is still the third leading cause of death in our society, despite antibiotics, despite vaccines, despite health care. About 20% in our society still die of infectious diseases. So the big killers in infectious diseases, the big three we call them, are one bacterial, that's tuberculosis, one viral, that's HIV, and one parasite, that's malaria. And these are the three big ones, and each of them kill 1 to 2 million people a year. Back in 1969, the U.S. Surgeon General stood up in front of U.S. Congress and made a very bold statement. And this is at the time when antibiotics were coming on, we had vaccines, and he basically said, it's time to close the book on infectious diseases. That's kind of like saying the Titanic won't sink. Got that wrong. So we know infectious diseases are outbreaking all the time, 1, 2 a year. We always see these things. And the best thing about that is microbiologists are never out of a job because there's always new ones coming at us. So you know, if you listen to the news, that currently there's concerns about a new influenza called H7N9 that has a high mortality rate in China. And there's also a novel coronavirus that's related to the SARS virus that is now going through the Middle East and is now being transmitted to France. Again, high fatality rates. How else do infectious diseases impact on us? Well, bioterrorism is a good example. And bioterrorism is really the use of microbes to cause terror in society. It's not really to annihilate and kill a bunch of people. It's just to scare society into a paralytic type effect. So it's more terror than bio. And bioterrorism has a history in what we call germ warfare. That's the use of microbes in terms of warfare. And this is an age-old history of warfare, and this actually goes back for example, when they were sieging cities in Europe and England, inside castles, etc., they would actually catapult plague victims into the cities to unleash disease in them. Or they'd snake in late at night and stuff corpses down wells to contaminate the water. And in a dark day of history in North America, the British actually took smallpox scabs and put them on blankets they were given to the First Nations, which pretty much completely decimated the First Nations because they had no resistance to this disease, unlike Europeans. So you all remember the anthrax scare in 2001 when somebody put anthrax spores in envelopes and dropped them in mailboxes in the U.S. Nobody saw this coming. Everyone thought that they would, spray spores from an airplane over a city or something. No one conceived this. And then if you know anything about the postal system, the envelopes go through automated machines. They're always squishing the envelopes. And this then released spores, infected some postal workers. And these letters were addressed to U.S. Congress people, and some of them got this. And there was a massive amount of antibiotic use. And then people stopped mailing them, I guess, and really that went away. So another concern we have with bioterrorism, as we learn more and more, if we sequence these microbes, could these things actually be, say, synthesized bioterrorism group? And can we make new and radical, scary germs to this? And hopefully it's not done, but there is a concern. We know that we can synthesize viruses that are very similar to smallpox. We can put these things together in the lab, and it still is a concern. So with all these concerns, what have us microbiologists been doing? Well, we've been studying these microbes, of course. There was a very famous microbiologist named Joshua Lederberg, and he basically said, it's our wits versus their genes. We divide in 25 years. If you're really good, maybe 20 years. Microbes do it in 20 minutes. There's no way we can outdivide these things, so we have to outsmart these things. And we've learned a heck of a lot in the last few years about how these microbes work and actually how these cause disease. And I could spend hours telling you about this. I'm not going to, but I am going to tell you a little story about E. coli 0157. And this is the organism that caused the Walkerton outbreak. And what we've learned, this microbe actually comes along, and it's got these grappling hooks that hook it down onto the intestinal surface Intestinal surface looks like a shag rug, and these things called microvilli, and it grabs on there. Okay, it's stuck, but that's just the start. These things are then so clever, they actually make this molecular syringe that then goes from the bacteria into the intestinal cell. And then they start pushing bacterial molecules right into that intestinal cell. And these molecules have all sorts of reprogramming effects on the intestinal cell, and they change all sorts of things. One of those molecules we discovered actually goes up into the intestinal wall and inserts there. Then the bacteria does something even cleverer. It actually retracts that injection system, and then it docks onto that molecule it put into the membrane. So it actually compresses right down to it. And say, okay, we're done there. We have to be. No, bacteria is still even smarter. They then grab this thing called cytoskeleton, which is mammalian proteins, and these things start to polymerize underneath the bacterium. And the bacterium then uses this as a force. And this then starts to drive the bacterium up on the cell surface, and the bacterium rises up on top of the cell surface. And then, if you actually look in disease, you see these microbes sitting up on these pedestals, we call them, sort of ruling their little kingdom, where they secrete the toxin and cause disease. So we've now learned all that. We actually understand how all this actually works. And the applications of this, I will tell you about in a moment. What about antibiotics? Well, you know that these are molecules that kill bacteria. These are the wonder drugs. I would say these are the biggest medical discovery in the 20th century. Ever since Alexander Fleming realized that these penicillin spores killed bacteria on plates. And these antibiotics block essential bacterial processes, and they weaken the cell walls of the bacteria lyse, for example, or they stop it from growing. And these are just miracle drugs. And, late World War II, the concept that penicillin could prevent gonorrhea, for example, and all these other life-threatening diseases were just major, major shifts in society. But like all good things, we can't leave them alone. So we use antibiotics for everything. And that we've overused these things, and that, of course, has repercussions. So you've also probably heard about antibiotic resistance. And that's the ability of these microbes to resist antibiotics. And pretty much as soon as antibiotics were discovered, so was resistance. You've also heard about these things called superbugs. These are microbes that are resistant to various antibiotics. Things like methicillin-resistant Staph aureus, or MRSA, or VRE, vancomycin-resistant Enterococcus. And these things are increasing in our society. And the problem with that is we don't have the antibiotics to treat them because they're now resistant. How did bugs become resistant? Well, they're very clever. And when you and I are born, we have a set of DNA. When we die, that's the same DNA we have. No matter how hard you try, you can't go and acquire DNA. Bugs can. Bacteria can. And so I liken the gut to the genetic internet, where they can download pretty much any gene they want. So they scurry around the intestine. Oh, there's an antibiotic-resistant gene. I'll just grab that one. And they become resistant. Or they can grab virulence factors. So microbes are always changing. And so as resistant genes are selected for by the use of antibiotics, so many microbes become resistant. And pretty much every bacterium we look at has much increased levels of resistance. Well, let's just make new antibiotics to get around that problem. No. The pharmaceutical companies are pulling out antibiotic work. It's very hard to make an antibiotic these days. And they've been trying, and they basically exhausted the low-lying fruit. And really, there have been very few new antibiotics. The number of approvals of antibiotics are plummeting. No one makes them anymore. Besides, you have a pharmaceutical company, you have a responsibility to shareholders, and you're going to give a drug for someone that uses a week and then they get better, or give someone a blue pill that makes them happy all their life. What are you going to choose? Lifestyle drugs. So that's where they've all gone. So we really are facing some significant issues here. Well, vaccines. Let's talk a bit about these things. These things are by far the best tool for controlling infections, and they work. And these are extremely effective. We've eradicated smallpox. There is no more smallpox in this world. We're just about there with polio. It's very close. Just a few countries still have small amounts. But again, there's been some bad stories associated with vaccines, and once in a while I have adverse side effects. A kid gets a fever or something. Unfortunately, there was a scientific fraud story published where they said that the measles, mumps, and rubella vaccine, which is a highly effective vaccine for these diseases, caused autism. Turns out it was scientific fraudulent, but that didn't stop it from going on the web, and that didn't stop many, many anti-vaccine people embracing it, such as Jenny McCarthy. She's a former Playboy bunny, and she appears on Oprah and promotes how terrible these vaccines are. And this has just had an awful effect in terms of the acceptance of vaccines. But we're fighting back. For example, just recently, Ottawa has passed rules that school kids have to be vaccinated or they're suspended from school. And so when vaccines work, they're absolutely delightful. Well, maybe that's not the right word, but they're terrific. So we talked a bit about E. coli 0157, how it works. I want to show you now how you can actually use that knowledge to make something. So E. coli 0157 basically hangs out in cows and doesn't do anything. The cows defecate this bug in the environment, either the water or a contaminated hamburger, and then it infects humans, and that's where it has the effect. So cows carry it, humans get the disease. You'll remember last September that XL Foods in southern Alberta had this massive beef recall because it had E. coli 0157 in the beef. This is the largest beef recall ever in Canada. 1,800 beef products were recalled. 1 million pounds of ground beef was composted. That's like 1,200 cows, and 800 workers were laid off. There were 18 human cases associated with this disease. Luckily, no one died of it, so that was good, and eventually reopened. Well, how do we use what we learned about all this fancy molecular knowledge to actually do something? So this is a childhood disease, and I was thinking for a long time, how do I make a childhood vaccine? And that's very difficult, and you always worry about secondary effects and everything. And then I was running out in the UBC endowment lands one time, and literally the light bulb came on. Why don't we vaccinate the cows for a kid's disease? If there's none in the cows, the kids aren't going to get it. And it was one of those sort of ice cream in the forehead moments, so we set about doing that. And so, but the problem is I know very little about cows. So I actually turned to a collaborator in Saskatchewan that actually said, well, why don't we try it in cows? And so we started immunizing these cows with those molecules I showed that the bacteria actually adhere to these things. And lo and behold, it actually worked. It blocks the cows from sharing this 0157. It works great. It takes the 0157 levels to below the levels you see in the winter when you never see 0157. So as a scientist, you say, I'm done, right? We got the vaccine. Well, not so quick. It's about $3 a dose. You've got to give them 2 doses, so it's $6. That's too expensive for feed these days. The other thing is, who's going to pay for it? The cows don't get sick, so the farmer says, well, why should I vaccinate my cows? They're fine. It's a human disease. There's also the people that said we should irradiate all the food, and this will work for the foodborne things, but only half of all 0157 is in the food. The rest is in water. And if you've ever eaten irradiated lettuce, you will know what I'm talking about not being good for all foods. So Canada spends about $200 million a year on healthcare costs associated with 0157. If we vaccinated every cow, the 12.5 million cows in Canada across the country, that would cost you $50 million. So do the math, you save $150 million. Great idea. Not yet. There's different government agencies that plays in all sorts of different political games, and so even though we've developed a commercialized E. coli vaccine, it's only being used in a small number of cases. Well, okay, so we've been talking about infectious disease. We are making progress on vaccines, antibiotics, and other ways. And if you look over the last 50 years of human history, we've been on this major campaign where the only good bacteria are dead bacteria. Kill them all because that's going to get rid of infectious diseases. And it has. You can look at many of these diseases, and pretty much most infectious diseases are going down. Very, very few are actually going up. So, but as in all wars, there's secondary effects and costs. And we're seeing something very remarkable. Yes, in the last 50 years, infectious disease has gone down as we clean up water and food supplies. But all these other sort of autoimmune type diseases are just skyrocketing the last 50 years. Asthma, type 1 diabetes, autism, multiple sclerosis, inflammatory bowel diseases, Crohn's disease. These are just going through the roof. How come? Genetically, we're going through, two generations in 50 years. It's not because we've had this mutation go through our population. That's just not possible. It's something else. And so the concept I want to talk about tonight is maybe this war on microbes is having a secondary effect and is actually impacting on many of these other diseases that we just really didn't think. So that takes us back into the world of microbes and microbiota. So microbiota is a term. It used to be called normal flora, but flora means flowers to some people, and so it doesn't get used anymore. It's a term for the bacteria that are in and on us. Remember I told you Anthony van Leeuwenhoek looked in his mouth to see all those microbes? Well, there's no way as a geek scientist I could believe someone that did an experiment 350 years ago, so I had to do it again late at night. So late at night I swabbed out my cheek, and I then put it on a slide and looked at a cell, very sexy cell, and saw all these things stuck to it. Of course these are microbes. And you can do the experiment. You don't believe me, you will see lots of microbes in your mouth. So you can stain these microbes, you realize they're there. So Anthony Van Leeuwenhoek realized they were there. But we've really kind of ignored them. We said, yeah, they're there, but we don't really know what they do. Let's not worry about these things. The concept I want to tell you tonight is going to really tick you off. You're more microbe than you are human. And I tell my students this, and they say, yeah, sure, Dr. Finlay. I says, well, okay, let's have a look at this. If you add up the number of human cells in you, then you add up the number of microbial cells in you, the microbes outnumber human cells by a factor of 10. It's 10 times more microbes on you than human cells. Now here's where we've got to get gross. 1 gram of feces, that's 1 centimeter by 1 centimeter, a little cube. That actually contains more microbes than the entire world's population of people. There's 100 trillion bacteria every bowel movement going to waste down the toilet. Told you we were going to get gross. What about genes? Bacteria have actually 100 times more genes than Homo sapiens in an organism. So that's a huge genetic potential that allows these bugs to do all sorts of things that we can't do. So what does this do for us? Well, basically it's a mutualistic type relationship where there's benefits to us, the host, as well as the microbiota. From a microbiota point of view, the bacteria benefit from nutrients. We feed these things 3 times a day. We water them, drink all the time. We're warm. We're 37 degrees. Who doesn't want to live in 37 degrees? We're moist. And this is really microbial nirvana if you're in the gut, except for the light at the end of the tunnel. So where are these microbes? Well, they're basically on surfaces that are exposed to the environment. If you go deep inside the blood and the tissue, you won't find these things. But the upper respiratory tract, ear, nose, mouth type thing, there's lots of microbes there. They decrease in numbers as you go down to the stomach, but then they increase in numbers more and more, and the large intestine is where the vast majority of these things actually hang out. So they're kind of on the surfaces. They don't really live inside. What do? Well, this is where in the last 5 years we're just finding stunning findings of what the role these things actually have in our life. For example, the immune system. That's how we fight off infections. We now realize that microbes play a key role in developing the immune system, and we'll talk about that in a bit. Brain development. We're now learning that the metabolites the bugs make in the gut go into the body and actually enhance brain development. Of course, metabolism. You eat your hamburger, what you had for dinner tonight. These microbes are digesting your food. You're not. You leave it up to the microbes. And the microbes also provide key vitamins to these things. We also know the microbes are critical for developing good intestinal barrier when you actually are developing. So they have all these effects. Well, how do you study these things? Well, there's ways of doing it. And the big question comes, if you believe all I've told you about microbes, the question is, well, are they essential for life? And so I sometimes ask my students, how could you test this? And they say, I'll just give massive doses of antibiotics. You cannot make a person sterile that way. But what you can do is birth them sterilely. There was a kid named David Vedder in California. Before he was born, they realized he had no immune system, so he couldn't live in the world, period. He would die. So they basically burst him sterilely by a cesarean section, then put him in a container. They called it a bubble. He became known as the bubble boy. He lived to age 12. Well, besides the fact that you're confined for a bubble the entire life, the life was not normal in many other aspects. He had many developmental issues. They had to give them key vitamins because he didn't have these microbes. But in the lab, you can do similar experiments, and these have been done since the 1950s, where you take an animal, birth it sterilely, and by C-section, and then put it in a container and always autoclave its food and water so it never sees microbes. These things are called germ-free animals. This is a misnomer. There's not germs. They're basically microbial-free animals. Germs are a subset of microbes. And so based on this is how we can learn some of the effects of what these microbes are doing, and such as the poor immune system, the poor intestine, and brain problems. You may have heard of this major project called the Human Microbiome Project. And this is take a whole bunch of people, sample a whole bunch of orifices in these people, and basically look at what microbes they are. And some of you might remember I actually introduced Craig Venter when he gave this talk a couple of years ago now. And Craig Venter was famous because he sequenced the human genome. not only a human genome, his genome. And many of you that saw Craig will know that Craig was very proud of that fact. So Julian Davis, a famous microbiologist at UBC, went up to Craig and said, Craig, that's really great, you sequenced your own genome, but you only got 10% of the genes. You missed the other 90%, the microbes. Craig sort of always up for a challenge. So we then started sequencing the microbiomes. So there's been a furious effort lately to sequence these microbiomes. The Human Microbiome Project also came out of the Human Genome Project, because when they finished the sequencing the human genome, they had all these people in these machines, and they sort of looked at each other, what do we sequence next? Someone said microbes, and we'll have jobs forever. And so they've been burning away at all these microbes, and the amount of data that's being generated is just stunning. And, we're learning all the sequences of all these individual microbes, their entire genetic component. But what we're also learning is that we are unique. Each of us has a unique set of microbes. We originally thought there's got to be a core of microbes that are in all people. We realize that's not true. There's some that are more common in most people than others. But the idea that there's a certain identical set in all people, that's wrong. These microbes are very diverse, and really we're each individual in that sense. So we are all different in terms of our microbes. So there's been a real struggle. Can we define a core gut microbiome? Well, there's a European group that looked at a bunch of Europeans and said, well, based on this, we can sort of come up with 3 kinds of microbiota. People have one of 3 different kinds of microbiota. They call these things enterotypes. And that was nice because then we thought we could pigeonhole different people into different enterotypes. And it's a nice concept, but really it hasn't stood the test of time. And really it's a continuum between these 3 types rather than individual species. One type, the Prevotella, named after a bacterium that's common in them, seems to be more associated with some diseases than others. So they generally cluster in these kind of general groups. Well, let's return to our story of how these microbiota can be involved in health and disease. So think about how you raise a kid these days. Kid drops a soother on the floor. When I was a kid, it got stuffed right back in my mouth. Now it's, oh God, no, we got to go autoclave that thing. You can't possibly, Oh, don't eat your lunch on that. I have to wipe it down with an antibiotic, wipe. Oh, wash our hands in antimicrobial soap because goodness help us if we encounter microbes. So this has led to a concept that we call the hygiene hypothesis. We are living too clean now. We've done our darndest to get rid of all these microbes, kill them all. And as a result, when you're young, you don't see the microbes. And then later in life, you see them, and you react to them. You have immune reaction to them, etc. So the concept is maybe we've cleaned up our world so much that we don't develop normally. Think how our great-grandfathers lived compared to how we live now. For example, in Japan, you put in a credit card in the automated teller machine, it actually sterilizes it. But then, what do? You pull it out with your fingers, which are coated in microbes, and they forget to tell them that fact. But, we've got this phobia of microbes, and I'm trying to argue maybe that's having some problems. So what about some of these diseases, and what are we doing? We know that obesity is just skyrocketing in developed countries, and developing countries, it really hasn't changed. How come? Well, you argue, easy calories, etc. But Jeff Gordon is a microbiologist in St. Louis. He did some very profound experiments. They're gross, I warn you. He takes the feces from fat mice, feeds them to thin mice, they gain weight. He takes the feces from thin mice, feeds them to fat mice, they lose weight. He takes the feces from heavier set people, puts them in thin mice, they gain weight. Stunning. He's only changing the microbiota in these things. And, there's a lot of compelling evidence. What we've also learned is the microbiota from heavier set people are incredibly efficient at generating energy from the food they get. They can just churn the energy out. Whereas the microbes from thinner people, they actually suck. They're not nearly as good at generating this energy. So maybe there's something to be said about this. But there's also diet concerns. If you eat a calorie-rich diet, of course you're going to enrich for microbes that can do this. It'll be different than someone that eats lettuce all the time, for example. So there are differences there. Inflammatory bowel disease is a very famous disease, obviously, and it has a big effect. And really, this is just inflammation in the bowel, and this leads to Crohn's and colitis, 2 inflammatory bowel diseases. And for a long time, we've known this is somehow microbiota related. There's been a big push to say these certain bacteria cause IBD, inflammatory bowel disease, but no one's ever really been able to prove that. There's a lot of microbes that are associated with these diseases. And again, it's kind of a chicken-egg type thing. If you have inflammation in your intestine, you will shift your microbiota. That makes sense because it's going to change the environment. But is it the microbes in the gut that are causing the shift in inflammation, or the inflammation causing the microbial shift? What we do know that IBD is really caused by these microbes somehow getting through the gut, then triggers the inflammation in the gut, and the body then hyperreacts to this, and that leads to these diseases. Let's turn to my favorite disease, diarrhea. Why study diarrhea? Well, A, it's fun, and B, it's still a major cause of disease in this world. There's 2 to 3 million deaths worldwide due to diarrheal diseases, mainly in children in developing countries. Very humorous side. It's very sad in that sense. And I'm sure many of you had a fine holiday at a nice beach resort where you're looking out at the nice sandy, warm beach, and you're sitting on the white throne saying, I'd like to get out there. So we see effects due to traveler's diarrhea, of course. So our lab has been working on E. coli. I've told you some of the E. coli stories already, which killed several 100,000 kids worldwide. And the other bug we've been working on a lot is salmonella. And I'm sure you've all heard about salmonella. And this causes 2 diseases in people. It causes typhoid fever, which is a systemic fever you get in developing countries. And other salmonella cause diarrhea. And it's thought that it causes 1.3 billion cases of diarrhea worldwide every year. So obviously it has a major impact there. So, we've been studying the Salmonella E. coli for a long time, and we started studying the human immune system. And then we went into the laboratory and basically challenged ourselves to think outside the box. And the question came, well, what about the microbiota? I wonder what happens to the microbiota when you get diarrhea. Neat question. I wonder what happens. So the idea was we could then take feces from, say, a normal mouse and put on a filter paper and stain the bacteria green. And what you see is basically a microbiologist's dream, a thing of beauty. All these long rod-like shaped things and all these little balls and everything. That's what the feces of mice looks like. As I said, a thing of beauty. So what happens if you infect this? Give it diarrhea. This is our E. coli model of diarrhea. Well, like every good scientist, you make all these bold predictions. You get diarrhea, so you've got to decrease the number of bugs. That makes sense. And then I thought all the bacteria would all go down equally. They basically all decrease in numbers. And seeing as it worked on a pathogen, of course the pathogen would just take it all over, and that'd be the only bug you see. And that's actually what happens in cholera. Cholera is another diarrheal disease. It causes 20 liters of diarrhea a day, very profuse diarrhea. It's pretty much a monoculture of the single Vibrio cholerae bacteria. But we wanted to look at these E. coli disease and ask what happens. So we stained all these bugs. We knew what it looked like before. And then we looked after. And what did we see? Well, we saw a decrease in numbers. We got that one right. The other 2 we got wrong. What we found is that all the rod-like bacteria seemed to stay more than little round ball bacteria. So that argued there was a shift. We call this a dysbiosis, an upsetting of the balance of the microbes. And then we actually stained the microbe that's causing this disease red. It's E. coli-like disease. And we only saw 2 little red dots in this whole field of vision that were actually causing this disease. So obviously there's something going on there. The question becomes what? Well, another puzzle we'd had, and I'll give an analogy. Let's say you go to a wedding, and 100 people at the wedding, you all eat the potato salad. It's got salmonella in it or something. 20 people get the squirts, the rest don't. Question becomes, how come? And if I asked you, well, if I wouldn't ask you this on the way in tonight, but if I asked you that, you'd say, oh, well, there's differences in the human genes causing this. And this is the same thing that's true in the animal models we use to study these diarrheas. We know there's some strains of mice that always die when they're infected. We call these things susceptible. And there's other strains of mice that are always resistant. And most gene mappers for years have been trying to identify mice genes that give you the resistance or the susceptibility to this disease. Sometimes they find these genes, but there's a whole bunch of cases they haven't. And in the E. coli diarrhea, they also haven't really found the host genes that are causing this. So, back to the lab. Let's do some wacky experiments. Let's actually think about changing the microbiota. Now we have to talk about fecal transfers. I know this is on all your minds. That's the only reason you're here tonight. We'll get there. So the experiment becomes, how do we take a mice and change the microbiota? We could feed it antibiotics to shift it, and we did that, and that caused infection and changes infection. But what we did is we took the feces from a resistant animal, and then orally gavaged it into susceptible animals. And we asked 2 questions. Could we change the feces in the susceptible animals to look like the donor, the resistant animal? And if we could do that, could we actually then infect it and change the outcome of infection? So we did the experiment. Lo and behold, it worked. It worked beautifully. If you put resistant animal feces in a susceptible animal, it's resistant to disease. We didn't change the mouse. We didn't change the infectious agent. We just changed the microbiota. You can do the experiment the other way. You take feces from a susceptible animal, put it in resistant mice. They're much more susceptible to disease. Those are profound statements saying the microbiota is really influencing these outcomes. So these fecal transfers are all the rage. They're very powerful, and you can do very cool stuff like humanize mice. So you take graduate student postdoc feces and you feed this to mice. You can take postdoc babies' feces and feed these to mice. And we could humanize mice. I mean, it's really neat because it opens up many experiments you actually can't do in people, though you can start to do these things. And if you're paying any attention to the news, you know that fecal transfers are all in the news these days. There's a terrible intestinal disease, very prevalent in Ontario and Quebec, due to the use of antibiotics. So the idea is if you're going for surgery, surgeon will give you a bucket of antibiotics so then they can hack away and you'll get infections, no problem, you come with antibiotics. But these antibiotics have profound effects on the microbiota. They basically kill off a bunch of the microbes. That actually kills off defense. So then this nasty pathogen called Clostridium difficile comes in, causes a horrible intestinal disease called pseudomembrane colitis, very tough intestinal disease. So how do you treat that? Well, they give more antibiotics. Well, this only works in less than 30% of the case because you've already wiped out most of the microbes. But it's been known for a while now, and it's really just been proven. And just last week, the New England Journal of Medicine, a great controlled trial study showed that if you did fecal transfers, feces from normal people into these poor sick people, you got greater than 90% cure rate. And the 10% that weren't cured, you gave them another fecal transfer, you fixed them. Stunning results. Scary when you think about the experiment in terms of what happened. But it worked so well, they actually had to stop the trial because it was working great. Well, then the FDA got in the picture, and they said, well, that's neat, but fecal transfer, we're going to call that an investigational new drug. And those are terrible words if you're in the drug business because it means a whole bunch of paperwork and a whole bunch of things, and really it's created havoc. You can't just go and shove feces into someone now. There's all these some other rules. So you remember last year in Burnaby, for example, they actually stopped fecal transfers. And there's a reason for it, because you remember the blood scares with hepatitis, et cetera. We don't know what we're transferring. How we get around this, I'll talk in a bit. But for any of you that are CBC fans and watch This Hour's 22 Minutes, there's just a hilarious skit on the concept of fecal transfers, and they even come up with a number you can call to donate feces. So you got to have a sick sense of humor when you work in this. Okay, microbiota and the immune system. This has just really radically reshaped how we think about immune system. I'll tell you this story. There's these two camps of immunology. You always see this in science. There's two camps of scientists, and there's a line in the sand, and they just butt heads all the time, and that's what science is all about. And they say, I'm right, you're right, I'm right, you're right, and basically this is it. And they were fighting about this one kind of cell called the Th17 cell. This is a key cell in the immune system. And one said, these mice have it, and the other said, no, they don't have this. And they were headbutting over this. We then teamed up with a group in New York, Dan Littman's group, and we showed very simply that it depended on the microbiota, whether or not you had these key immune cells. Not only that, we showed that it depends where you bought your mice from. One mouse supplier, they had these cells, one mice, they didn't. So you go back and do all these papers these groups are publishing, you just basically look at where they bought their mice, conflict resolved. It was due to the microbiota and where they bought these mice. So then they could do fecal transfers and fix it, etc. And we're seeing this again and again. Regulatory T cells, another key immune system that basically balances your immune system. This depends on some species of microbiota. And this has profound effects on how our immune system develops. So that's going to affect vaccine responses, for example. Vaccine responses in developing countries are very different than developed countries, maybe because the microbiota is different. What about mothers? If they have group B streptococcus, they're given antibiotics at birth, high doses antibiotic. That's good because it prevents the transmission. But what's it doing to the kid and the kid's microbiota and the mother's microbiota by giving these antibiotics? Okay, on to another disease that I never thought had anything to do with microbiota, and that's asthma. So we know asthma is going through the roof in developed countries, and it's not changing in developing countries. And the big question is, how come? And there's all sorts of concepts about this, but we know it's becoming very prevalent. Well, what about a role for the microbiota in this? There's actually a whole bunch of, I call these things smoking guns, that indicate the microbiota may have a role in this. So when I was a kid, there was one kid in my school that had one of those asthma inhalers. And because there's only one, he was different, he had to be a mutant. These days, a quarter of all kids have these things. You know, a playground, kid runs up slide, comes down, puff, 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 hands a puffer to his friend who takes puff, puff, he goes up slide kind of thing. I mean, it's humorous, but gee whiz, I mean, this is just phenomenal disaster in terms of kids and stuff. So we know that the gut microbiota is different between allergic and non-allergic infants. Well, that could be just because different immune responses, but there's an association there. We know that if you get antibiotics in the first year of life, you increase the rates of asthma just by taking antibiotics in the first year of life. We know that you can give some probiotics to maybe decrease some allergic diseases such as eczema. We know that things called short-chain fatty acids, which are products of the microbiota, are different in allergic and non-allergic infants. And here's a statistic that just stunned me. If you were born by a cesarean section, you have a 20% higher chance of getting asthma than if you're born vaginally, just because of the microbes you get on the way out, which I think speaks volumes about this microbiota concept. We know that living in farms, these awful dirty places where there's all these microbes, actually get much less asthma. So what did I have to do to asthma? Well, my wife Jane is a pediatrician, and I was excitedly telling her, you know, we give these antibiotics, we shift these microbiota, we change diarrhea. And she said, you know, Brett, give these antibiotics in the first year of life, they have much higher rates of asthma. And I said, oh, really? I said, well, she's my wife, so she has to be wrong, right? So, but I've always learned, listen to your wife. You know, I go and read, of course she's right. Wives are always right. And it was true. And so I thought, wow, well, but then who's done the experiment? Nobody's done the experiment to prove this. There's all these correlations, but let's see if we can't prove it. So there's some very useful animal models, again, to study asthma. There's a mouse asthma model. And what you do is you take this mouse, and then you immunize it to something called ovalbumin, just a chemical. And then later you give intranasally down into the lungs this ovalbumin, and it reacts to it. It's an allergic reaction, which is basically allergic asthma. And this is the model everyone used. So I went over to Kelly McNagny's lab here at UBC, who has a model for this mouse asthma. I said, Kelly, can we treat mice with different antibiotics and affect the outcome? And so we did that experiment, and lo and behold, we found profound differences in the asthma levels depending on which antibiotic we used. So we've now gone into several experiments. We know that it's just in the early period of life that this microbiota is so critical for shifting everything. And you can probably guess it's related to something to do with the immune system developing in this period. So where do we take it? I mean, curing mice asthma is not exactly going to make you rich. How can you do something about this? Well, we're now involved in a child cohort study, which is 3,000 children across the country, where we're taking feces from birth up to age 5, and then looking at which microbes are associated with wheezing and not. And we've learned which microbes in the mouse are associated with these things or not. And then we can do experiments like take allergic kids' feces and non-allergic kids' feces, put them in mice, and then study this whole thing. So you can see where this might go, where you could actually have this sort of probiotic cocktail. You could look for kids. Do they have the right balance? If not, can you rebalance it so they have much less chance of getting this disease? Microbiota and autism. What the heck could microbes have to do to autism? Well, we know autism is increasing like all these other diseases I've talked about. I've also told you we know from germ-free animals that microbes are involved in brain development. A large percentage of autistic kids also have gastrointestinal problems, which is the microbes in the gut, so you can imagine there might be a correlation there. Now, there's a big controversy right now whether or not these things are involved in autism. There's some groups say yes, some say no. But I want to tell you one story that just had a profound effect on my interpretation of this. And this is a colleague of mine. It's a true story. She had a friend who had a kid, and this kid was normal. At 18 months, needed antibiotics for a different type of infection. Kid became severely autistic. Mother was convinced it was microbiota. Doctor said, no way. Mother said, no, put him on different antibiotics, shift the microbiota, and let's see, do the experiment, basically. Doctors didn't want to do that. She finally persevered. Doctors put this kid on different antibiotics, shifted the microbiota, and this autistic kid got better. They stopped the antibiotic. The kid became autistic again. By now, the mother's absolutely convinced this kid's got to have a fecal transfer, but no one wants to do a fecal transfer in a kid. So she went to a doctor that does fecal transfers, and he said, I can't do this in a kid. She persevered and got the guy to do the antibiotic experiment again. He did. Antibiotics. Autistic kid became normal. Take off the antibiotics. Kid regressed back to autism. By now, he's convinced. So we then, at 6 years of age, took the mother's feces, put it in that kid. Kid became normal. Kid is normal ever since. Kid is working in a microbiology lab as we speak. And so, now yes, that's an N of 1. And autism, as we know, is a whole spectrum of diseases. But, based on stories like that, you got to believe that in some cases of autism, there is a connection there. Aging. That's a topic of subject in our house these days, What could the microbiota have to do with healthy aging? Well, remember those different enterotypes I told you about? It turns out that as you age, your microbes tend towards the disease-rich sort of type of grouping. Once you turn 65, your microbiota actually shift towards what we kind of think is the unhealthy type of microbiota. So when you're first born, your microbiota bounces around, then you have your childhood microbiota, and then you go into puberty, and puberty, hormonal change, etc. And then you basically get your adult microbiota, and that lasts the rest of your life, unless you do something drastic, like drink a lot of Guinness beer or something, and that shifts your microbiota until you're 65. So there's a lot of studies now going on that we know that aging people actually have different microbiota. Whether that actually corresponds to healthy or unhealthy aging, it's still seriously under research. Well, I've told you, I hope I'm convincing you these microbes are important. And there's this thing called the disappearing microbiota hypothesis. Maybe we're actually killing all these things off and they're becoming extinct, and we actually can't go back the way our, say, grandfathers and great-grandfathers did. So the average child gets 10 to 12 antibiotic doses before it reaches at the end of its childhood. And we know that antibiotics have drastic effects. Streptomycin, metronidazole completely shift the microbiota composition, as well as we're killing off all these microbes. So the idea is that, maybe that we're driving some of these things really to extinction, and, we really can't, recover these microbes unless we do a road trip to Africa or ask Grandpa to donate something to us. There's significant concerns there. All just being aside, we know that the more diverse the microbiota are, the healthier you're going to be. And as you decrease diversity, you increase disease. And so all these things we're doing with microbes, you wonder, is that actually impacting on us? What are we going to do about it? Well, we're actually making pretty good progress in growing these things. Inside our gut, there's no oxygen. So you can't just haul out a bacteria and plate it on a plate and then grow it. E. coli you can because it actually tolerates oxygen. But most microbes out of the gut, they die the second they see oxygen. So you have to grow them in these fancy hoods where there's no air. These are called anaerobic hoods. The other thing that we've come to realize is this is a community. The idea of taking a single bacterium out of its environment where all its nice neighbors are and put it on a plate and ask it to grow doesn't work. And so we've learned things like you can take organic solvent extracts of feces and add it to bugs, or you grow them as communities. And M. Ellen Verco is a professor at Guelph, a good friend of mine. And boy, if you think I've got a sick sense of humor, you should see her talk about that. She grows feces. So she takes these feces, put it in bottles, and grows it anaerobically, and she's cleared out the whole floor because it stinks so much. She calls this thing the robo-gut. And one of the things she did with the robo-gut is, remember I was telling you about C. difficile and the fecal transfer? She actually took these microbes she grew in here and put it in 2 patients. She calls this repopulating. And she repopulated these 2 people, and great, it worked. So you can see where we're going in the future instead of fecal transfers if we had defined cultures. She was shut down because the lab wasn't clean enough. It wasn't like a drug, so she's now gearing up to more like a pharmaceutical type production. But you can imagine that's where you can go. Also, all the molecules these microbes make, this is just a tropical forest of molecular diversity. Emma calls this liquid gold. There's all sorts of molecules in there that do all sorts of neat things that we're just starting to play with and understand. So, obviously my lab works on infectious diseases in developing country, and one of the interests we had in the lab is, could we have a developing country mouse? Well, how do you do that? Well, one of my grad students made the neat observation, well, we eat differently, obviously, than people in there. Let's just change the diet. And he did this thing, and lo and behold, it actually shifted more towards the developing country thing. You can fecally gavage feces in there, like kids in developing countries get, and he's actually got some really neat models now to study diarrhea, study vaccine response in developing countries, study malnutrition, chronic diarrhea, and again, all just by shifting the microbiota. Where are we going? What's the future hold? Well, things like RoboGut will become online. We will start to grow these things. The idea of fecal transfers, I think, will not be used very often. this thing called metabolomics. That's mining all the small molecules these microbes make that talk to our bodies. This is just a profound growth area in the future. What we need are intelligent ways to modify our microbiota. We're pretty good at figuring out what microbiota we have, but how do we actually tweak these things? Can we go in and surgically take out or add particular microbes to these things? Personalized medicine. You talk about you'll have your genome sequenced, the doctor will say which drug you should have. They got it wrong. They forgot about microbes. So if you think about it, you take a drug, and adverse drug reactions, for example. You take a drug, who breaks that drug down? You don't. Microbes in your gut do. Who forms all the secondary metabolites that cause these adverse drug reactions? You don't. Microbes do. So this whole thing about personalized medicine, adverse drug reactions, they're going down the wrong route. I'm convinced of this, that these microbes are actually playing a role in this. And we should actually be typing the microbes in us as opposed to the tiny differences between humans in their genetics. So drugs of the future will be things like probiotics, but it won't just be one lactobacillus in yogurt. This is going to be gelatin-coated capsules of a whole community of bugs, for example. There's things called prebiotics. These are sugars you can feed people that enrich for certain types of bugs. If we know which way we want to enrich, we can think about that. And we're all experimenting with sort of novel therapies, immune modulators, phage therapy, etc., ways of modifying the microbiota. We're not there yet. I think we're at the stage where we can look what are sick people's microbiota and healthy people, which things we think are associated with it, but knowing that, we're still not at the stage yet, because it's really a new field. It's only in the last 5 years we've come online on this. So, this is a really exciting time. I think this is one of the biggest revolutions in biology in the last while, and I think it has profound impacts on how we think about human health and disease. So finally, with all this in mind, I want to close with the concept that we should really be nice to our microbes, and they'll be nice to us. Thank you very much.
Hello, everybody. I'm Michael Serapio, as I put on my jacket. Anyways, I'm with CBC News Network, and I'm going to be moderating the Q&A, and actually I'm going to ask you, Brett, to sit over on that chair over there. I'll turn on your microphone as well. Excuse me. And we have two stands set up, one at either side of the theater, so the lights are now focusing on them. Feel free to stand up and we'll kind of take people's questions back and forth. We also have people on social media via Twitter asking questions, which I'll read to you as we do this about 40-minute Q&A here. So, Brett, the fecal really hit the fan with you. Kind of fascinated there, my friend. I want to begin with the starting point as the ideas that you present tonight. The idea that microbes, more than genetic sequencing, can lead to cures of diseases and disorders. How popular is this idea now?
It's just starting to gain traction. I think the appeal of it is that if it's very hard to correct a genetic defect in a person, but microbes are pretty plastic and you can muck around with them pretty easily without having profound effects. So there's a lot of thought that, in terms of gene therapy, maybe microbes would be a better way of doing it. But the fact that they're going to be the panacea and the cure-all to many diseases, I don't think that's going to hold true. But really, we're just starting to, infect other fields, shall we say. People in other fields are just starting to realize these microbes play effects. And, immunologists embraced this fact fairly early on. Now even developmental biologists starting to realize this. And really, it's just the tip of the iceberg as people are starting to realize this.
When you talk about the disorders and the diseases that can be traced to microbes, and obviously there are things that we grew up with already knowing, things like the bubonic plague. But you talk about, and you brought up one example of asthma, the difference between vaginal birth versus a cesarean section. What other diseases are tied to this lack of exposure to microbes?
Well, the joke in the field is what isn't. And I think the pendulum swung a little bit too far in terms of microbiota causing everything. But We talked about obesity, we talked about autism, I didn't talk about type 1 diabetes, for example, multiple sclerosis, there's hints at it. Pretty much inflammatory bowel disease, asthma, so all these kind of autoimmune self-reaction kind of things as opposed to infectious diseases. There's several examples in cancer, intestinal cancer, et cetera, being associated with microbiota. But again, this is only in the last few years, and it's just an explosion. We're really kind of just finding our way.
Oh, we have a question there. Please introduce yourself and feel free to ask your question.
My name is Elias, and what about cancer? I'm curious to ask you something about that. There is a doctor I learned about recently in Italy, Dr. Simoncini, and he has a fungal hypothesis. Of course, he was fired, but that doesn't really mean much. But what do you think about that? I'm sure you know about it, right?
Yeah. Obviously, you got to prove it scientifically. I think that the concept of cancer, even the BC Genome Sequencing Center has now found bacteria associated with certain types of cancer. It makes sense because these microbes produce all these molecules that go throughout the body. Cancer is really just cells that start to divide, and any way you uncouple their regulation, you can cause cancer. So some of these molecules can be doing it. Some of the bacteria, like Helicobacter pylori, which causes stomach cancer, that does it by basically causing so much inflammation in your stomach, you're constantly turning over these cells that eventually get mutations that lead to gastric cancer. And it actually is classified in the same as a carcinogen, same as a cigarette, that glass. So I'm not saying all cancer are caused by microbiota. Obviously they're not. We know there's many cellular defects associated with these, but there will be a subset that microbes will play a role in, and we really just starting to learn that.
On this side of the room, again, please introduce yourself.
Hi, my name is Erica Kubanek. I'm wondering about what you feel the role of supplementing probiotics in a more complex other than just the single lactobacillus, and also if you think that diet, lifestyle, those sorts of things can create a favorable terrain for a healthy microbiota.
Yeah, I think the idea of a probiotic being a single bacterium is a bit naive. I'm sure in the future that probiotics will have multiple species because we need many of these things. We know that, what is a healthy microbiome and what is diet. We know that has profound effects on the microbiota. Like if you go on a diet, within 3 days your microbiota is heavily shifted towards actually the healthier kind. The studies have been shown we lock people up and feed them on diet and not, and they actually know this. China did an amazing experiment. They took an entire village and moved it to a city, because you can do this in China, and gave them sort of McDonald's coupons equivalents, and showed that it had just profound effects on their microbiota. And so we know diet has a major, major impact. The microbiota of you with someone you're living with, for example, will actually be more similar than your identical twin living on the other side of the world. So environment plays a major role in what your microbiota look like.
I want to do follow up on that, though, just because it does raise an interesting question. You talk about pharmaceutical development, but how much of this can be, preventative? If, for example, we say to our child, you know what, go roll in the dirt. Go stick that in your mouth. Call me later.
Well, I think that's a very good point. We have this germ phobia, so, and even I think the Bible says eat a peck of dirt before you die. There's a reason for that. Obviously, you have to have some common sense, and, it has had a major decrease in infections. We saw, for example, when we had, the flus, swine flu, et cetera, that, we convinced people to sneeze with their arms in here. And that had a profound effect on many different aerosol-type diseases. So, I think that, environment can play a major role in these types of things.
To the other side of the room.
I was wondering whether the increase of autism could somehow be could be affected by the records kept of autistic people. Like some kids in the past may not have been diagnosed, even though they had autism. Like whether record keeping has become more efficient and stuff.
So as opposed to just microbes itself?
He mentioned the increase of autism, and opposed to just microbes.
Well, I think there's been a huge increase in autism, much of because of diagnosing what previously wasn't called autism. And I think there's a lot of discussion, what is autism? And I think autism is a whole spectrum of diseases. I don't know anything about the medical records. I don't work in that area, so I can't comment on it.
Dave Patrick, UBC School of Public Health, and you'll recognize me as your trumpet player. Brett?
You know what? No.
Next question, please.
No, it's
Thank
true. Have you given some thought, or have you heard of anyone giving some thought to how Koch's postulates would be altered or modulated to deal with a polymicrobial etiology?
Yeah, well, I'm not going to say that's a good question, but it's a really
you could be nice
point.
and call it a good question.
Well, so remember I told you about Koch's postulate. There's the rules to say a microbe actually causes this disease. I think that some of the stuff I was showing about the microbiota really profoundly, impacting on the outcome of disease, that has to be taken into account. And really, we're only just starting to think, maybe we should think about that if you're trying to say it only causes disease in a subset of people because they have a subset of microbiota. Yeah, it's a good point, and no one's doing that yet. I mean, TB, it infects everyone, so we don't have to worry about this. But some of the other ones that might have a certain subset of the population, they should be written for that.
Oh, go ahead. And again, please introduce yourself.
Hello, I'm Corky, and I'd like to ask Dr. Finlay, what would you say the government policy and regulation should be if different from now?
Regarding fecal transfers or microbiota in general, or what?
Microbes in general, or your research or general research in this area.
Yeah, I think it's far too early to start regulating right now. I think the world of probiotics needs a bit of regulation because right now probiotics are not considered a drug, so you can make any claim you want, it cures whatever, and you're not held to it. I think that as they start to go through FDA like clinical trials and treat it as drugs, we'll be able to regulate those a little more carefully, and so the natural medicines will have much more of a drug basis to them. Right now, in terms of rules, we barely know which microbiota is good and bad, so it's just way too early to think about any kind of regulations. I think, although we do need to think, about, say, massive use of antibiotics as growth promoters in animals, which we do. That's not a good idea because we're decreasing the microbiota, et cetera. So it's just too early.
Tim Ma. And Brett just asked your thoughts on this. In the course of your presentation, you mentioned 3 enterotypes, if I recall correctly. I immediately thought to traditional Chinese medicine, and in traditional Chinese medicine, there are at least 4 basic body types that have been identified. And you feed, or depending on your body type, you do well eating oranges or you don't do well eating oranges, etc. I'm just curious: is there some cross-pollination in terms of scientific investigation and drawing on some of these traditional medicines?
Yeah, I don't know the answer to that, but I think anything like diet, for example, that will get you different microbiota. Eat oranges or not, that's going to change microbiota. I don't know if that correlation being drawn with the 4 Chinese types. I don't know.
To the right.
Hi, Samantha Gray. Just wondering about fermented food. Around the world, cultures have always eaten fermented food, which we tend not to do in our culture today. And just wondering the role that might be playing in disease progression over time.
Yeah, there's some hints that actually fermented food is good, and by not eating it, there are issues with it. Again, strictly from a scientific basis, I don't think we know enough about, you're getting these bugs and not those bugs, so therefore it'd be healthy or not. So I mean, this whole field is really, really new and really swirly, and we can't say that, yes, you must eat fermented foods because you have this bug, it'll do this for you. That's where we're heading, and there's a lot of work that's going in this area, but to try and sort it all out. And as you know, studies in people, eating one fermented food, well, what'd you drink last night? That could impact too. So human studies are very difficult to nail down the one cause that does this.
Sorry, if I can interrupt, would you then say that really the knowledge that we have is more relational than causal?
Yeah, right now it's just correlations. Microbes and disease people look like this, microbes and healthy look like that. But whether the microbes are causing disease, we don't know that. We're heading that way, and in some cases, like asthma stuff, it looks like it, but we haven't got there yet.
Okay. To the left.
We know, my name is Inga, we know empirically now that animals that are fed genetically engineered foods, corn, soy, that they will develop inflammatory bowel problems, that they do get degenerative disease in the major organs. And we also know that the antibiotic-resistant bacteria and viruses that are used in the genetic engineering process release into the gut and therefore give you a new microbia. Now, is it possible then that the genetically engineered foods dramatically shift the microbia?
I mean, genetically engineered foods, for example, pesticide-resistant plants. I don't subscribe to that theory, I'll be honest. I'm not entirely convinced, and the data's not that strong. For example, I mean, I'm an avid Diet Coke fan. Well, that's a GMO because aspartame is made in E. coli, so that's why I know I'm weird. But, you know, genetically modified just means you've manipulated some gene. We've been doing that for generations with foods, but now we're actually inserting genes in, like flavor savored tomatoes, for example. That just affects how they oxidize, so the tomatoes aren't as ripe. So to make broad blanket statements about GMOs, I don't think the data is that strong.
Sir, is the mic off?
No. But if
I
you
just
want
to counter something,
yeah,
and he has
just
said.
do follow up.
Yeah, it is not true that the genetic the hybrids, etc., that we have used through a natural process are in any way identical to inserting the gene, which upsets the entire synchronism of the entire gene thing. So there is a tremendous difference between the two, and you can't say we've been doing this for God knows how long. The genetic engineering that we have now is relatively new.
Okay. Did you want to?
Not really.
Okay. So let's move on to the next slide then.
I didn't think I deserved it.
If you can you just tip the microphone up so we hear you?
Yes, sorry about that. My name is Nick Alexander. Thank you for coming to speak tonight. You mentioned a shift in the microbiome around age 65. In the rest of your talk, you didn't speak too much about similar shifts from anything sort of coming from within the body. Can you say a few words about why that shift and why it seems to happen at that time? And in your discussion, it seemed a little isolated.
No, I think there's a very good reason. As you turn 65, your immune system wanes, basically gets weaker. And I think we and others have some data in the lab that really the immune system seems to be sort of controlling the microbial balance. And if you then weaken it, which we know is 65, your antibodies go down, your vaccine response goes down, that's also going to impact on your microbiota control. And I think that is probably the underlying cause for that shift. You don't maintain it the way it was. But during your life, anytime you do something like change diet, if you get stressed, for example, you shift your microbiota, all these environmental things have profound impacts on your microbiota, which could have downstream consequences.
Sorry, I'm going to jump in before you ask your question, only because I do have a question here from social media. This is from someone on Twitter by the name of Mara Dat. Would it be plausible or useful to map the genomes of all human microbiota? If so, how could it be done? And I think you touched upon that in the lecture, but if you can expand on that.
Yeah, I mean, that is the stated goal of the Human Microbiome Project, is to sequence all the microbes you can in a human. Now, there's two ways of doing it. One, you can sort of use this tool to sort of generally get an idea of what types of microbes are there. The other way is just to sequence absolutely everything there. You don't know which genes they come from. Or you can grow them and then sequence them. So this is a major project underway. It's generated a phenomenal amount of information. It's been very frustrating to get useful information out of it because we generate these gigabytes and terabytes of data, but we don't know what it means. We still, anytime we sequence an organism, only have an idea of maybe what half the genes that organism do based on similarities to other organisms. So we're really stuck, but there's a major effort to try and sequence all these things, and eventually we'll start to figure out what they do.
Is it possible to do it all? I mean, when you talk about just the number of microbiota that's out there.
Yeah, I mean, sequencing, I mean, it's just getting profoundly quick. And yes, it's possible, but why would you do it? I'm not entirely sure we're going to learn all that much from the exercise. We will get a catalog of all these genes, but most of the genes don't have a clue what they do with anyway. So I'm not sure that's necessarily, that should be the first goal of microbiome work.
Hi, Joshua Chambers. I was just wondering, with all this new research and everything that you're doing, I find it very fascinating, and I'm just wondering how that relates to us going to the doctor and having these conversations when we now have stomach problems or family members that have stomach problems, and are in terms of diagnosing these sorts of things. What does this say for the doctors, and are they diagnosing for the wrong things, and are they overlooking this kind of research right now? And so that's my concern.
It's certainly becoming on their radar, and people are coming in. People read the web, and they come in with the doctor's office with this. And physicians are certainly aware of this. But right now, autism or asthma or whatever, you wouldn't send a kid out to have their microbiome sequence, because if you did that, we wouldn't know what to do with it. So from a medical point, we're not there yet. It's coming, and I think in the next 5 to 10 years, this whole idea, I think personalized medicine will include typing the microbiota of children, for example, and say, oh, you're at risk for these things. Maybe we should do something about it. But it's still far too early to walk into a physician's office and say, I want a fecal transfer to cure my baldness. It's just not there yet. Or I would have done it. So it's coming, but I think there's no doubt the medical profession is certainly aware of it, and the lay public are certainly. There was New York Times this week, a huge article on it. Everyone's becoming aware of it, but then how to use that information, for something useful, it's not there yet.
Again, introduce yourself, please.
Robert Young, Urban Barnes Foods. Dr. Finlay, I'd like to say I really enjoyed your talk. I wondered if you had any information on organic produce and the use of organic pesticides. What percentage of farmers that grow organic produce use pesticides? That's the first question. The second question is, what is the effect of either chemical or organic pesticides on the good and the bad bacteria?
Yeah, I can't comment on your first one. I'm not an organic foods expert. I don't know that one. Again, like I say, anything you do in the environment will shift microbiota. Now, we all eat food, and we know that people living on farms, for example, they have different microbes in the city. So I'm sure, depending on what your foods, if it's coated in cow feces or not, it's going to be different in terms of the microbiota. But what that means in terms of health, and is this good? Is organic food better or not? Organic, I don't know. I think that the jury's really still out yet. We just don't know enough. Does this mean anything? Is it significant? We don't know.
Hi, Heather Thompson. Question about the availability of fecal transfers in the more near future, while they're still developing products like Repopulate for people that have been diagnosed with irritable bowel disease or Crohn's disease and are currently suffering, and you possibly have a healthy donor that could donate to that patient?
Like I say, the rules just changed. Up until a week ago, there were physicians both in the States and Canada that were doing it, but most of them didn't want to brag about it or tell about it. But like I say, with the bringing in of this new investigational new drug FDA ruling, it has put people on the spot, so you can't do it. I do know that on YouTube, there's lots of videos how to do it yourself. It's technically not that difficult. Like everything. But so right now it's in a state of flux. I agree there's some studies just starting on inflammatory bowel disease. There's a couple of promising studies that maybe fecal transfers might work. I do know if you have Clostridium difficile, if your choice is dye or fecal transfer, it's a pretty simple answer as far as I'm concerned. So they're sorting it out, but it's just in a state of flux as of this week.
Is that the same in Canada and the U.S.?
Is that the same in Canada and the U.S.? Well, pretty much whatever the FDA rules, Canadian standards use a similar standard. So, there's a lot of discussion what we should do. And I think there's a real motive now to get away from fecal transfers into defined cultures as soon as we possibly can. For a whole bunch of reasons, because we don't know what we're transplanting, all the viruses and things you worry about finding 10 years down the line, some virus or oncogene or whatever it might be.
Hello, my name's Roxanne. Good evening, Dr. Finlay. I do want to point out before I pose my question that infectious disease, the mortality rate for infectious disease plummeted well before vaccination across the board. Measles, I have the stats for BC, plummeted well before vaccines. And scarlet fever is a good case in point because there was never a vaccine for scarlet fever. Even the British Medical Association in 1928 reported...
Can we ask a question, please?
Yeah, it pointed out in 1928 that there was more smallpox in the vaccinated population than the unvaccinated, so I can bring that documentation to your attention. Also the fact that Andrew Wakefield's work on the MMR vaccine and how it affects the gut has been duplicated.
It's fraudulent. It's been shown
not been fraudulent. It's been
Next
politically...
question, please. I don't want to go there.
Can we ask a question,
No, I'm going to ask my question. It was politically smeared. It was not fraudulent. It's been replicated. The question is, are you aware of any of the following studies? University of Purdue...
Okay, if she's going to ask her question, can you just hold off on the applause, please, because we should hear the question up here to answer it fairly.
Yes. Okay, all of these studies compared, in the case of University of Purdue, vaccinated versus unvaccinated dogs, University of Pittsburgh, vaccinated versus unvaccinated primates, and Kigs out of Germany, large survey, compared vaccinated versus unvaccinated children, and across the board, the unvaccinated were healthier, had less asthma, had less allergies.
So your question
diabetes.
is?
Are you aware of those studies or the work of, for example, vaccine developer Bonnie Dunbar, who's pointed out all the studies?
Thank you. Are you aware of those studies? Seems to be the
B
question. I'm sorry, ma'am. Your question, please.
Are you aware of all of these studies? Are you aware of any of these studies?
Thank you very much. So that's the question. Are you aware of these studies?
I've seen some of them. I really don't want to go there on this. I mean, I think this is a public forum on the use of microbiota. It's not on vaccines. The vaccine area is very contentious. I know there's some very strong views in the field. I know the scientific literature in the vaccines, and in my opinion, vaccines, the evidence is absolutely overwhelming. They're the most effective public health measure we have. I don't want to go there. Thank you.
Okay, to our next question, please, on the left.
Tough follow-up. I'm John Davies. I have a question about diversity. So we know, like, for humans, genetic diversity is really important in our population against disease as a species throughout time. Do you think having microbiota diversity is important throughout the population, and that's why you see all these different, I forget what they're called, enterotypes?
Yeah, the enterotypes. Yeah, that's a great question. That why do we have this diversity? I mean, if humans are 99.99% the same, why isn't our microbiota? I think part of the problem is I think it's actually microbiologists' problem, how we categorize these microbes. Because we, as microbiologists, like to say, this is this species, that is that species. And we classify these things. But if you actually take a look and say, okay, we need 10,000 genes in the gut for the microbes to do, when you look at functional gene types, actually the diversity is pretty similar. And there's this redundancy between microbial species. But the data does show, in terms of health, the more diverse microbes there are, the healthier you are. The impact of this diversity on human evolution, I've just written a couple of grants on this, actually. I mean, it's a neat area, but nobody knows anything yet. And could it be affecting evolution? And what is the impact of these microbes on evolution? It's a great area, but we know nothing about it. Yeah, good question.
The other side of the room.
My name is Leah Moy, and my question is about whether you can comment about what you talked about, the role of microbes on adverse drug reactions, and perhaps give specific examples where they might play a bigger role than genes.
Yeah, the question is adverse drug reaction. So we know that in a population, if 100 people take this drug, it'll work in 75, and maybe 5 will have these awful drug reactions. The question is why. And so in science, they've been looking at, what are the differences between our genotypes, you and I, and there's these things called single nucleotide polymorphisms or single base pair change between individuals. And there's some data show that in some cases it's just the differences in the genetics of the people. But if you think about many cases, they can't find differences. And so my proposal is that when you take a drug orally, it goes into the gut and the bugs break it down. Not you, your bacteria, and your bacteria are quite different than my bacteria and anyone else's. And so they then form these secondary metabolites. And in a subset, if you have certain microbes, they'll form this, say, adverse chemical that then you react to. So I think the simple idea that if you just have your genome sequence, you can detect whether or not you'll be susceptible to certain drugs. I think the microbes are being ignored, and I think they may play a large role in many of these adverse drug reactions.
Do you have any specific drugs in mind that you think that might be the pathway that's working?
This is really early days. I mean, even in personalized medicine adverse drug reactions, there's just a couple of examples coming out where we understand what the differences is to give you differences in drug reactions. It does bring up a bigger question from a pharmaceutical point of view. If you're going to get a drug approved, should you test all the microbiota in the people that you're getting it approved in? Should you be looking at this? And that's a very scary regulatory question. Nobody wants to go there, but it is being talked about.
Hi, Dr. Finlay. My name is Paul Lithgow. I'm from the Genome Sciences Center. You've talked a bit about how things like antibiotics and diet and exercise can modulate the microbiome composition. Do you have any examples of how the human genome can affect the microbiome composition?
Yeah, beautiful question. No, not yet. So, no, the question is, okay, is our genome actually shaping the microbiota, or is, it just depends on who you encounter in life. And just an off-the-cuff thing, environment, I would say, probably affects about 75%, genotype affects about 25% or 2. So we know, for example, if you have inflammation or stronger knockout animals that lack inflammation, they have different microbiota. So there's host genes there. But it's a fascinating area, but it's very complex. You have to overlay the human genome on the microbiome, which is a major bioinformatic issue to try and sort it all out. And no one has really seen through that area with any kind of logical answers yet. It's definitely a growth area, though. Stay tuned.
To the right.
Hi, my name is Odette Slater, and I'm not a scientist at all, but my question, I think, sort of relates to just the last one. I'm wondering if there are implications here for genetically inherited illnesses like Huntington's disease, and if yes, is there any research being done in that realm yet?
No data. Good question. Just don't know. I mean, I'm not sure it'd be Huntington's disease, but inflammatory bowel disease, I would guess, might have something to do with it. I mean, we know there are genetic links to several diseases, and if those genetic links might affect microbiota, you could argue, well, maybe that's driving it as opposed to the particular, effect yourself. So again, I don't have a good answer for this. It is a really good question, and I think people are starting to look at this, and as people realize it's a genetic disease, but we can't find a genetic defect or don't know what it does, maybe we'll embrace the microbiome. Maybe in a subset of those cases, they'll be affected by the microbiota.
Thank you.
Hi, Cynthia Davis. It's Michael Pollan, the Sunday New York Times.
Yeah, it just came out.
Yes, and a book and a movie cannot be far behind, so this is just such a timely talk. He says eat a wide variety of everything, mostly raw. That's his stuff, eat lots of varieties of food. Anyway, my question is, and you may not be able to answer it, if a woman has to take antibiotics during a pregnancy, now that's during the pregnancy, can that affect the baby after it's born? I know it's supposed to be sterile.
Yes, we know that the microbiota make molecules that actually go across the placenta, across the blood-brain barrier, the placental barrier into the fetus. And there are some studies that show that antibiotics taken in the mother that shift microbiota have effects on the development of the fetus and then the animal that's born. So yes, there is concern. I mean, antibiotics, when you need them, are wonder drugs, but we tend to abuse them. I think it may be causing problems. And so, again, it's early days, but there is data to indicate that, yes, indeed, and we know from animals, giving antibiotics to the mother will affect, the young later.
And I'm just going to point out, for timing purposes, we probably have about 3, possibly 4 questions left. We have 3 speakers up at the mic. Let's go see how we get through the last 3 here. Yes, sir.
Yes, my name's Kevin Smith. I'm not a scientist either. I guess my understanding of what you were saying is that there were a lot of autoimmune diseases where it appeared to be the microflora kind of had a role to play in that. But I'm wondering if there's any kind of a hypothesis or speculation as to what actually happens, because the body's kind of going against itself because there are not all the germs there or all the microflora that was there before. And what is the mechanism or speculation about what the mechanism of the body is? Why is the body turning on itself?
Well, early in life is where you set up yourself for the rest of life. And that's where you learn to tolerate, things so you don't become allergic later. And the general concept is that if you don't experience these microbes early in life, you're not tolerant to them. And should you encounter them later in life, you then react to them. So inflammatory type reactions might be cause of it. So the idea is that there's a real window early in life that's when your microbiota really, we know it shapes the immune system, it shapes other ways you develop. And if you don't have that set correctly, like, Th1, Th2 balance in asthma, for example, then that screws you up later in life. So the idea is it's what bugs you get early in life, and that's setting you up. And you don't get them done right early in life, it's going to affect you later in life with these autoimmune type diseases. That's our current hypothesis. It's not proven. There's a fair bit of data pointing in that direction, and we know that's one of them, but there could be other ways too.
Yes.
Hi, my name is Lalo. I actually have a question on the technical end of things. I noticed from your slide that it says that it's new technologies that are giving us unprecedented insights into health and disease. And I know, just to draw an analogy, that in particle physics you need a bigger and better atom smasher to get closer and closer. And I'm just curious what the new technology is. Is there some sexy new toy that's the atom smasher equivalent, or is it just as boring as the broader better
It's
program?
just the ability to sequence phenomenal amounts of data in a real hurry. It's also the ability to basically amplify ribosomal genes to then figure out what these microbes are without having to grow them. But really, it's just the power of sequencing. Because before, you had to grow these microbes on plates, and you can't grow these things anyway, so we really couldn't study them. So it's really the sequencing that's unleashed this whole world.
So there is no poo smasher.
Well, you can find anything in our lab. Not going there.
Thank you. I'm Najib Ayas. I had a question about vaccines. Actually, no, I'm just kidding. But very, very provocative talk. I guess one of the questions, I just want to get a sense from you in terms of the future. Like, obviously, what you want to be able to tell somebody is to say, these are the 5 foods that you should eat to make your microbiome very healthy, and these are the 5 foods that you should avoid. Do you get a sense that we're close to that point? Is that, quickly on the horizon, or do you think that, it's just things are so complicated we may never get there? I just want to get a sense from the standpoint of health from that, view.
Well, we do know that, high fat, high salt diets shift the microbiota towards the unhealthy types. So generalities, but and we do know some foods shift microbiota different ways. But whether you eat one particular, bran flakes or whatever it might be, people like General Mills that make foods are very concerned about this and trying to figure out how can we make more nutritious foods that the microbiota can then utilize more nutritiously. But to do it intelligently right now, nowhere near. I think that's way down. I think right now the best advice is just eat a well-balanced diet, exercise, and that will keep your microbiota nice and healthy.
Go. Yes, sir.
Given your caveats about not knowing enough yet, what's your views on, let's say for a geriatric person using something like a VSL number 3, I don't know if you're familiar with that, very strong probiotic, daily, should they be stopping it whilst they have antibiotics? Should they be taking it continuously at all, or periodically, or only on certain symptoms? Or what do you...
Yeah, probiotics by definition are safe. And there's no adverse drug reactions with probiotics ever shown, so they're safe. So that means you can take it, it's probably not going to hurt you, sort of thing. My personal view on probiotics is that they're too simple right now. The idea of one strain, like the VSL strain you're talking about, is going to cure everything that ails you. I think that's naive. I think probiotics, the future will be complex mixtures, or including other ways of modulating diet to give significant mixtures. But by definition, probiotics are safe, so if you want to take it, it isn't going to hurt you. In some cases, it may help. All the trials on probiotics, looks kind of promising, but they're not overwhelming wonder drugs that cures everything that ails you, like some of the claims are made.
So I want to take this opportunity, Brett, to thank you very much for an incredibly interesting and sometimes humorous talk. And I want to thank you, Michael Serapio, for moderating this session. I want to thank the Vogue Theatre staff and my own staff, Bernadette Ma, and all the other staff for making this evening a success, CBC and the Georgia Strait for co-sponsoring. And most of all, I want to thank all of you for coming, because these conversations, I think, are very important, and stay tuned for our next one. And please join me in thanking Brett.